Okay, so it's really a pleasure and honor to introduce Bruce Hungate. I will keep this very short and sweet so you have plenty of time to listen to Bruce. So Bruce is a professor of biological sciences and director of Ecosystem Science and Society Center at Northern Arizona University. For those early career researchers in the audience, I just want to say my first talk that I heard from Bruce was as a postdoc, and it was just inspiring. It was one of those talks where you hear some amazing science and you just think about all of the wonderful ways that microbial communities interact with one another, with the environment, and it was almost like poetry. So Bruce is a very, very leader, very influential person in the field. And I just encourage everyone to sit and enjoy and really soak in all of the amazing science that you're going to hear about as our first keynote. So thank you.
Thank you so much, Emily. What a kind introduction. And thank you all for being here. Wild microbial ecology. To me, the wild adjective there is redundant because ecology is wild, right? It's the interactions of organisms with their environment, with each other, in a community, in a complex ecosystem. But in microbial ecology, there's a good reason to lean a little bit on that adjective, which is why I put it in the title. And I really like to think about it. I get it. Friends of mine give me a hard time for that adjective, but I think it's good. And it takes me back to my first introduction to microbial ecology. This is my very first paper ever in Jim Staley's lab at the University of Washington, where I had 79 isolates, pure cultures, isolated from desert varnish. The desert varnish is black because it's made of manganese oxide, oxidized manganese. And we were searching for the microbes that we thought responsible. One of the debates was, is it an abiotic processes that forms this varnish, or is it biotic? And so in microbial ecology, what you do is get the organisms out and look at them by themselves, which is fantastic when you think about it, because we can't do that with people. We can't do that. I guess we have gnotobiotic mice, right? But it's really hard. With microbes, it's standard. And it's a great way to do ecology because you can create the environment that you want to look at in the lab with just the organism you want to look at. I remember my very favorite of these strains was a Geodermatophilus. You have to love the name, right? Earth skin lover. It just loves to be in like desert varnish, right? And it had these pink mounding colonies that were really fun to look at under the dissecting scope. And I will never forget adding this drop of reagent. I don't remember the name of it, but that turned the pink colony dark blue, indicating the presence of oxidized manganese. This was a manganese oxidizer, as were, I think, 75 of the 79 isolates that I found. So I went with this finding to Jim. It was a pretty quick assay. I could do all the cultures in an afternoon. I was really excited. We had proved that bacteria were creating this desert varnish. And if you know Jim Staley, he smiled and nodded. Yes, 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 that's very good. But we can't actually say that in the paper, because this is a lab study. We understand it from the lab. So to me, that moment was important in my career, because from that time, I really turned towards, before we get there, the point Jim was making is what happens in the lab might be different from what happens in the field. And of course, we need those lab studies to really understand what the organisms can do. And we need the field's context to see why it matters, because wild microbial ecology is super important for, things like organismal health. If you've got the right microbes, that makes a huge difference as to whether you're a healthy human or sick. Or food production, the agriculture, the microbes that we need to grow plants efficiently. Or in my favorite area, that's a weird way to talk about global climate change, right? But it's an area in which I put a lot of time and effort. These microbes produce and consume gases that warm and cool the planet. And they do that in the wild, in nature, which is really, to me, where I wanted to study. At my core, I'm an ecologist. And so being outside and studying the organisms outside was really an important driver. But I look back at this time because, then I went on to do my PhD with box and arrow diagrams like this and soil microbial responses there all lumped together. There was no more 79 isolates. And of course, we know that's a terrible underestimate of the actual diversity that's there now. We had to lump them all together. But we also know that those microbes produce about 60 petagrams of carbon per year in soil as CO2. So that's petagrams of carbon as CO2. It's a lot. And in order to understand the trajectory of climate change, we need to understand the ecology of those microbes that do it. So for me, it was a huge sacrifice. I was working with Terry Chapin at right here at Berkeley. And here, this is down at Jasper Ridge, Heather Reynolds and I injecting N15 into soil to get microbial uptake of N15. Heather was looking at the plants. The wonderful thing about this with isotopes is you can get the quantitative actual rates of uptake. Plant uptake. And Heather, I was super jealous. She could isolate each individual plant species and analyze it separately. For me, of course, we had to lump it all together into one compartment, the soil microbial responses. But it was quantitative. We could estimate fluxes in nature. We could think about how they're important for things like global photosynthesis, feedbacks to climate change, that I was super interested in. So this was a lot. I spent a lot of time on this. And I think, till about, 2010. Of course, the molecular revolution was happening. We all knew soils are incredibly diverse. And it was, I remember these discussions. It was actually painful to think, oh, you plant people have it so great. You can calculate these rates and measure them and know what they are and know the biology, the ecology of these organisms. For microbes, it was really hard. And then to introduce one of the most overused and hackneyed metaphors in all of science, we'll call it the box you can't see inside. Suddenly, we could, right? And what came out of that box or how we looked inside it was with omics, which is just a tremendous revolution in the science and showing all this diversity, all this metabolic capacity, all this wonderful science. And I resisted for a long time getting into this because the metrics they just didn't resonate with me. I'll use a simple comparison. It's as though, bread right now costs about $2 for a loaf, eggs about $4 for a loaf. And we know from that, eggs cost 100% more than bread, right? What if that's all that you knew? It's relativized. Do you know how much bread costs? No. Do you know how much eggs cost? No. And of course, you can do wonderful analyses with this kind of relativized information. But this, for me, I know it's simplistic and there are reasons why my view is wrong, but this is how I really saw a lot of the omics work. And really what's been my mission is omics is to say, wait a minute, I know it's hard. I know we might be wrong. I know there are biases, but we have to estimate those actual amounts. We have to get beyond this, of course, to ecosystem science, to quantitative ecology. Of course, that's just one lens to look through it, but that's really, as I'm thinking about my own science and what's motivated me, that's really been an important driver. So what I'm going to share with you today is very collaborative work from lots of different groups, many of them listed here, lots of different folks, and funding sources as well. And I want to tell you the story of the origin of QSIP, which really started with a transatlantic Skype call. This was in the days of Skype. Becky was doing her master's work and she was doing SIP. This was really my first molecular experience was with her thesis. And of course, with SIP, you have your centrifuge tube, and it's a density tube where you spin the DNA and it separates by density. You slice it up into lots of fractions, and then usually you pick a couple regions, analyze a heavy fraction and a light fraction. Becky was insistent, and she said, I want to sequence all the fractions. My first reaction was, that's nuts. It's so expensive. How are we ever going to analyze this? What are you talking about? And as the conversation went on, I became totally convinced, because what became clear to me and to Egbert and to Jane, who were also on the phone call, is that if we do this, we could turn CIP into a technique that estimates the molar abundance of those isotopes, not just an index of whether or not some was taken up or some are growing and some are not, but actually like a mass spec. It's just a compromise of 6 orders of magnitude of resolution, but it's like a mass spec in that you get atom fraction excess. So that to me was one of those moments of insight, because that's the metric of biogeochemistry when you're doing isotope tracing. Suddenly you can convert these measurements into rates of interesting processes in nature. So that is why I'm so excited about it, if you can tell. What else? This really began with Egbert's work earlier, noting that if we add 18O labeled water to any system, we can detect the who's growing and who's not. And he also noted who's dying and who's not, because if you look at the rate at which natural abundance DNA disappears, that's an independent measure of mortality, another, key part of ecology. So there's that. And then this is the basic idea of CIP here with 18O water, that you add it to a community and it gets incorporated into DNA. But really, I've tried, 5 or 6 different ways to explain QCIP. And my collaborator, Jane, on her YouTube channel has really done the best job. This is not it. I've already gone over all this, so we can just get to the fun part here. Jane created a metaphor using candy and crackers. This is a version of the peanut butter on a cracker leaf you might have heard in aquatic microbial ecology, where the idea is what the bugs in streams are really getting nutrition from are the microbes that are growing on the leaves. It's like peanut butter on a cracker. Well, that's another hackneyed metaphor. And Jane very wonderfully expanded it to be different kinds of candy on all different kinds of crackers, because really not every leaf is the same. And the candy are the different microbes. So we have 4 different taxa here put into a centrifuge tube. Watch how Jane turns herself into a centrifuge, spinning it around, and all of a sudden, the microbes separate by density. You should check out our YouTube channel. There's videos on there. It uses the Passover holiday as a metaphor for science. It's really wonderful. So these DNA from these organisms migrates in the tube according to their mass. And that mass with QCIP is what we can quantify. So what you can see here is the orange on let's see, that's on your right. That's the natural abundance control with no isotope tracer. And you can see on the tube on the left, it's exactly the same place. The orange ones did not move at all. They are not growing. Same with the blue. They did not shift at all. The yellows, you can see they shifted down the density gradient, so they're growing. And the pinks, sorry, I have to do this, they grew good and plenty. That's the name of the candy. Okay. Oh, I should have been doing it on that. Yeah. Okay. So there's her YouTube channel. You can check it out. With this technique, I'll show you examples today of the kinds of questions we can answer. They're super, super basic, like that first one. How fast do these organisms grow and die in the wild? How much do they compete? Food webs. How do microbial traits affect what they do in the wild? And really my favorite is, how can we attribute element flow, quantitative rates of element flow to individual species? And I'll say species here. It's almost a bad word in microbiology, right? We need to bring it back. It's okay, even if we don't know exactly what it is. So how fast do they grow? This gets into a model that Ben Koch helped develop that is all about getting DNDT, the change in population numbers over time. And what we can do with that from these various environments is end up with data like this, where each dot there, each space on the x-axis is an individual taxon, and you can see growth and death and the net between them. So what's important here to me is that y-axis unit. That's a growth rate, per capita growth rate. This is a classic metric in ecology. This is the foundation for a lot of theoretical ecology in population and community scales. And it's one that if we're interested in element fluxes, we can use to get to ecosystem scale. It also integrates a lot of physiology. So it's a super profound metric of life. We've looked at systems around the world, or around the country really, to so far to look at growth rates. And this is just a compilation of a lot of them. Over 100,000 growth rates, put it on a log scale, we can see it a little better. Average growth rate of bacteria in soil is about 0.01 per day. That's pretty slow. That's, they'll double in 100 days or so. But there's the answer to our first question. There's a lot more there to dive into, right? What's the distribution of that growth rate? How does it vary taxonomically? And we're getting some insights there. We can also look at things like competition, right? Here's evidence from a paper led by Steve Blazewicz that there's density dependence in these microbial communities. So as density goes up, growth rate goes down, which is the pattern you expect when there's intra-specific competition. The organisms are competing with themselves. As they get more and more populous, resources become less and less abundant, and their growth rate goes down. That's one possible explanation. Food webs. This was a really fun analysis because we looked at bacteria that eat other bacteria, like all these different taxa, and my favorites, Delovibrio and Vampirovibrio on the right. What that y-axis shows is the growth rate of those predatory bacteria compared to the average heterotroph in the community. So the take home here is that they grow fast. These predators are growing rapidly in natural soils. They're growing by eating other organisms. So predation is probably a pretty important structuring element for energy flow in these microbial food webs, and especially the Delovibrio and the Vampirovibrio, which are obligate predators. Some of these other groups are facultative. They can eat their colleagues, or they can eat other dead stuff, as bacteria do. Vampirovibrio is a fun one just because of the name. It infects its cells, inserting cytoskeletal fangs into the cell and shooting the DNA in there and doing its damage. Here it is on a green alga. And these predators grew especially fast in response to a resource perturbation from the bottom up. So give the base of the food web more energy, and that propagates all the way up to top predators. Here's an image we came up with. It's a metaphor. It's predation, right? Just like predation that we think of, wolves eating caribou. So what about traits? This is, of course, a hot area in microbial ecology now. What can we learn from microbial traits to predict how they're functioning in the wild? And to me, what techniques like QSIP give us is a benchmark to look at, the actual performance in nature. So here are just some examples. Some bacteria will grow fast, some grow slowly. Ember's done some really good work on this, showing, for example, compared to environment, that the genes that are inside the organisms have a stronger influence on growth rate than does the environment. And sorry, I forgot this point, that we're looking across 4 different ecosystems, 4 different climate zones in this analysis. So there's a lot of environmental variation, right? And we think of organisms as being responsive to their environment, and of course they are. But what Ember found is that that environment had an influence, but there was a far larger influence in who was there. And that taxonomic identity of the organism has a lot to do with predicting how fast it grows in the wild. To me, that's a profound conclusion that we've seen again and again and again, that who you are really matters. Junwei Li looked at 2 particular traits to get at the same question, starting with which genes affect growth. And here's a hypothesis I'm sure you've all seen, the idea that the number of copies of the ribosomal RNA gene should be a good predictor of growth rate. So bacteria vary in how many copies of this gene are in their genome. And that's important because it indicates the capacity to ramp up protein production rapidly, which is a function you would expect you would need if you're going to grow rapidly. And this is also pretty well known that in pure culture, what we see is a relationship like this. The more copies you have, the faster your maximum realized growth rate can be under laboratory conditions. When Junwei looked at this at first in the field, this is what we saw. Oops, it doesn't work. So, one interpretation of this could be, well, other traits are important under the conditions that we did this assay, right? It's not just maximum growth rate is maybe not a useful trait for predicting growth in nature when resources are super scarce. But we also found that when we add resource, we see the trait emerge. When we add resources, that slope gets positive. So we do see the relationship in soil. The intercept is much, much lower because they grow more slowly, but the same relationship is there. Bree Finley did some nice work looking at growth across a geosequence, different parent materials, looking really at the relationship between soil minerals and growth rate. And what she found is that as mineral content goes down, growth rate goes up, as though there's an interfering function of these soil minerals with bacterial growth rate. It's not too surprising. You can imagine lots of reasons why that would happen. But super fascinating in her analysis was that there were some groups that showed the opposite pattern. So just to orient you to this graph, that scale on the bottom shows the slope for each taxon of that relationship between mineral content and growth rate. So for most of them, it's negative. As mineral content goes up, growth rate goes down. But for this group highlighted with the arrow, all those little red spots, they're responding exactly in the opposite direction. So to me, this indicates maybe they're mineral tolerant, maybe they're just good at growing in that environment or less affected by the minerals than their competitors. Here's Megan Foley's, one of the bottom lines from her paper, again, was that knowing the identity of a taxon goes a long ways towards predicting its growth rate. So these are 18O compositions in 2 different sites, Angelo and Hopland. Each dot is 1 ASV or 1 taxon. And what you can see is if you know what the taxon is at Angelo, you can predict what its growth rate will be at Hopland, right? And vice versa. So again, who you are really matters. These are very different sites, different rainfalls, but who you are really matters. It's like a pine tree versus an oak tree. Those growth rates will be preserved even in widely different environments, or the differences between them. This is work in progress by Alicia Purcell, comparing field and lab QSIP assays. This one was very disappointing because what we found is that what we measured in the lab was really different from what we measured in the field. So talking about wild microbial ecology, Alicia did her work actually injecting the 18O into intact cores in the field and letting the incubation happen there. So that's where the microbes are living. That's where they're growing. That's a lot of ecosystem science. That's how we work. And of course, we also do lab assays. So this one was disappointing. I need to update it with new data from Ember that says it's really not this bad in another trial. But this is important stuff. Like, how do we scale from the lab into the field? How do we take that information from culture to rates in the environment? And then how much element flow can we attribute to individual species? We also worked on the same gradient experiment. This was Alicia's PhD work. She's since gotten actually a CSP support for her work in Antarctica. This is a great story because she wrote that proposal as a PhD student. So just a little plug for the awesomeness of the CSP program and JGI in general. Yeah, she landed that grant as a PhD student and is now working on her samples from Antarctica. For her PhD, she did this work in the field, focusing on this transplant experiment. The transplant was a climate change treatment. We started it in 2002, and her thesis work was in 2016 or something like that. So 14 years or so of experimental treatment. And I'll just show you the metaphor here. The control is A on the left, and the heated is B on the right. If you imagine the carbon content of soil as both the home and the food of microbes, they're happily filling all the niches on the left. But with long-term warming, what happens is there's a huge decline in soil carbon. There's a massive reduction in soil bacterial growth rate that makes it look like a more depauperate system. Bram Stone, now at PNNL, took data from the same experiment and built a scaling model basically to get that species attribution of flux. And this is the form of the model. You know, you've got growth rate, which we measure, CUE, which we infer, all kinds of parameters in there that we're estimating. And it really was one of those exercises just to show we can do this. And yes, this is almost certainly wrong, the rates we're coming up with, because we don't have a way to check them another way. That's what we need to find out, right? How wrong are we and how can we do better? What he found, again, just shown in this metaphor here, is that one taxon, this Brady rhizobium driving that giant machine underground, was doing the vast majority of the work. And we could actually account for more than 50% of the total carbon flux with 3 taxa in bacterial carbon flux. So it's a little bit of a different message from a lot of what we are super interested and excited about, all the diversity. In this case, when we were looking at function, we didn't need that many organisms in order to predict the whole. Not a conclusion I really feel that great about as a microbial ecologist, because we always want diversity to be super important. I don't know why, just it's cool. And it is important regardless of calculations like this, right? All of these organisms are important. But in this case and in others, we found that we can actually explain a lot of the ecosystem process with very few taxa. And just one note here. Oh, sorry, those are out of order. So this is a good place to say, yeah, let's see. There it is, the HTCIP, and I have the, oh, it's buried here, sorry. The new developments in CIP and QCIP are super exciting to me. The slide that Emily showed about the capacity here and the ability to estimate atom fraction excess for a MAG in metagenomes is wonderful because we can do the same kind of scaling. To me, that's the Q in QCIP. What it means is it's not an index. It's not something we just put in an ordination analysis. It's actually an estimate of a physical property of that DNA, the molar fraction of the rare isotopes, or how much isotope tracer has gotten into that sink in the system. So it's super important. It's not just a cute branding trick from me. QCIP means we're operating the centrifuge like a mass spec and estimating excess atom fraction. And that means, as scientists, we need to take that as far as it can go, I think. This is the ecosystem scientist in me speaking, which says, assumptions, bring it on. We're going to be wrong? Yep. We think DNA extraction is not sufficiently quantitative. Well, I'm sorry, that doesn't mean we can't estimate how much DNA is in soil. We have to. And when we look at that with an ecosystem lens, actually, this is a little bit of a side note, but in ecosystem science, we've done chloroform fumigation for microbial biomass. Have any of you heard of that technique? Just a few. Okay, great. Because you'll love it if you haven't heard of it. You take a soil sample, you expose it to chloroform. That lyses the microbial cells. And then all of them, supposedly. And then you extract it and analyze that extract for how much carbon or nitrogen and the isotopes, this is one reason I like it, is in there. You compare that to a soil where you just do the extraction to get all that background carbon. The difference is microbial carbon, divided by 0.41 to correct for extraction efficiency. And so this method, came on board in the 70s and 80s and has been tested and looked at all kinds of ways. We know it's wrong. And yet it's been very, very useful to probe how it's wrong, when it's right enough, what we can do with it. And so I think for one message that I'd like to give this field is go ahead and assume, go ahead and take that step into calculating a flux. I mean, that's my particular metric of scaling up because it's super interesting and important. And what's happening here at JGI is really exciting. It's just a collection of some of the folks who've been pushing forward these techniques and making all kinds of super interesting contributions. And then in the area of human health, I wanted to note Emily Cope has been working with QSIP in the gut of mice to understand the microbiome and Alzheimer's disease and other diseases. So there are lots of different applications of taking a quantitative approach to scaling up to things like mouse health or human health or global climate. So I want to wrap up with some thoughts about growth as really an integrator of lots of scales of biology. And to start with, all those instructions or guidelines in the genome that build cellular machinery that does metabolism with all the enzymes and substrates and energy. It's all about or it all integrates up to growth. And then when you think of interactions between cells and the external environment, matching that growth with the right stoichiometry. Here with QSIP, we can measure rates of ammonium uptake into cells, into microbial cells, and have some taxonomic resolution to partition that up. And that, from an ecosystem's perspective is super important because that nitrogen cycle, the amount that microbes take up into cells has a huge influence on plant growth. And that in turn has a huge influence on the global carbon cycle. We've seen that again and again in global carbon cycle models. So this is a great example, I think. Also, that cell needs to shoot out extracellular enzymes to break down complicated substrates and support the machinery to bring those into the cell to use in this metabolic system. It also has to deal with environmental stressors or the need to adhere to soil, to communicate with other cells in forming biofilms to withstand drought stress. So extracellular polysaccharide production coming out the back of this machine. And all of that to grow, to reproduce its DNA, a fundamental metric of growth, which these CIP techniques really get at, and make new cells. Another part of the external environment, of course, are the predators that will come and introduce the counterpart to growth, the inevitable outcome of growth, which is mortality. And then we can think of it in an evolutionary context and ecological, with organisms of different phylogenetic histories coexisting together and doing all of this generation after generation, growing and growing until there's some drought stress and the coxoid shaped spheres didn't produce enough EPS and it's not protected. So these traits of organisms that will have impact on whether they survive or not and keep growing. And to me, all of this really is in service of another grand scaling of microbial growth from those bacteria that sit on the hairs of roots in soil, the fungi along with them in a pedon in the tundra here, and how their activities scale up and influence processes on larger scales, and really all the way up to the globe. So with that, I'd be happy to take any questions.
Do we have any questions in the audience? Yes.
Oh, hi, Bruce. Hi, Bruce. So just wondering, like, which genes control the growth rate of the microbes? You mentioned seems like the genes, they explain a lot of variance of the growth rate.
That's a wonderful question. So as I understood it, is what accounts for growth rate? What explains variation in growth rate? And I did mention a couple of examples like 16S copy number. Am I getting your question right?
I'm just wondering, which genes, do you know which genes control the growth rate of these organisms?
No. Short answer, no. Yeah, and there's so many candidates and possibilities. I wouldn't even dare to speculate.
Okay, thank you. I'm very curious about that.
Yeah, me too.
Sure. So, Bruce, you talked about a lot of organisms, but you guys, another time,
Yeah,
and
yeah,
yes, their total number with stimulatory
yeah,
organisms that are using them might not get high.
yeah.
Does it matter if you can't capture those organisms who have massive outside influence on my this matters for it. How to think about this that you're likely not just you, all of us are likely
Yeah, it's a great question. And I think that I'd start with, I think it's a super exciting hypothesis and sometimes borne out. It reminds me of the rare biosphere argument. I mean, I think that's one way to put it. And I think it's important to honestly confront evidence to the contrary. That's point number one, like what Bram did with his analysis. For some processes, I think it's very likely that it won't be the case, that there are rare organisms that are really dominating it. And so I think, number one, we have to be careful because it's a very attractive argument. You know, the whole keystone species thing. How can you go wrong with that? That's so cool. And point number two, I think there are likely cases that we can imagine that they are important, even though they're rare. Like nitrification is a great example, right? And there, I think, there are a couple of things. I'm reminded of Katarina Pop's work on ammonia oxidizers, including ammonium oxidizing archaea. She used QSIP with mRNA to show a really nice correlation between the rate of production of the AMO-A gene in both bacteria and archaea and their rate of overall growth as inferred through 16S rRNA 18O uptake. So we can do it. And I think there is an argument there for not forgetting that amplicon sequencing can still be useful and powerful, especially when you have a targeted low abundance population that you think is important. The second thing, or the third thing, I guess I would add to that answer is QSIP with DNA and the philosophy that, I tried to articulate here, that it's really valuable to have that excess atom fraction estimate. Why not with the transcriptome? Why not with the metabolome? Why not with the proteome? Because we can take the same kind of scaling argument and apply it to all those systems. And yes, we're going to be wrong. Yes, we're going to have to make assumptions. But, imagine that all put together. And then you could get organisms that are not growing, but are very important. So I'd really love to see development there. I feel like that's a huge frontier in making this science scalable to the wild.
So I think my question is actually kind of related to that. In the example where you found that the Brady rhizobium was really dominating the carbon processing, does that mean that they're the only ones in that system that are really growing at an appreciable rate and everybody else is losing? Is it kind of a zero-sum game, or is it much more complicated than that? And in any given sort of average soil, even though one really doesn't exist, right? What fraction are actually growing at a rate that will allow replacement versus what fraction is dying?
Yeah, super interesting question. I think that, the first thing I would comment is that the relative growth rate, which is, like in units of per day or per time, cells per cell per day, that is obviously a super important, useful metric, but it's not absolute growth rate. And that Brady rhizobium was not the fastest growing taxon. So it's a function of relative growth and abundance to get the absolute carbon flux, right? That particular example, that particular metaphor, I should have said, was in response to resource addition. So that was the case when there were a lot of resources abundant. When it's more oligotrophic, it's the functioning is more even. Okay. Yeah, but I think it's a great question, and it reminds me that one of the risks of not or of using an index of what you think is an activity towards what you're really interested in biology without being quantitative and explicit about the assumptions you're making is that you can take something like relative growth rate and assume that that means, well, they must be growing the fastest, therefore the most important. Even if they're super rare. And so with some of our QSIP discussions go through that, encouraging, no, we really need to see it scaled up before you can make that statement, because just because it's growing fast does not mean it's important. Yeah.
All right, we have one more question,
Okay.
Bruce, from online, from Venkat. He's saying, Bruce, great talk. Does the QSIP technique work quantitatively to quantify microbes in oligotrophic environments? In other words, how much biomass do you need?
Oh, yeah. So we've had luck with QSIP in environments like, well, in Antarctica. This is a glacial sear. The youngest site is 60 years deglaciated. So that's a good example where the DNA was low. I'm not good with numbers like how much we need. Many of you here do, but we've been successful in extracting and analyzing DNA from environments where it's low in concentration. It reminded me of something else I wanted to say, but it flew out.